When a T cell encounters an antigen-presenting cell, it dramatically reorganizes its cytoskeleton to form a specialized contact site called the immunological synapse. The synapse is organized into 3 zones, the outer of which, called the DSMAC, contains a lamellipodium-like branched actin network formed by the actin-nucleating ARP2-3 complex. But, as John Hammer from the National Heart, Lung and Blood Institute in Bethesda explains, actin's organization in the medial PSMAC region of the synapse is less well understood.
We had shown in a previous paper by a former graduate student, Jason Yee, that if you image actin dynamics in a T cell stimulated on an artificial activating surface using F-Tractin, which is an indirect reporter for filamentous actin, you see this outer ARP2-3 dependent branched network, and then inside that you see these actin arcs. They weren't very clear in confocal images, but they were definitely there.
These arcs hadn't been observed using GFP actin, perhaps because GFP actin is a poor substrate for formin proteins, which could be responsible for nucleating arc assembly. To get a better look at the formation and organization of the arcs, Hammer and colleagues, led by postdoc Srish Murugesan, teamed up with Eric Betzig's group at HHMI's Janelia Research Campus to visualize the immunological synapse using Superresolution microscopy. Using 3D SIM and TURF SIM, the researchers confirmed that the PSMAC region contains concentric actin arcs that are decorated with bipolar myosin II filaments, forming a contractile network that moves inwards towards the center of the synapse.
Not only were these things much clearer in TURF SIM than we'd ever seen before, but what we saw was that embedded within this outer branch network were linear actin filaments or bundles, which appeared to be nucleated at the plasma membrane, and then the filaments drive down through the outer branch network and then bend and orient to create the arcs in the medial portion of the synapse.
The topology of these linear filaments nucleated at the plasma membrane suggested that they are assembled by formin proteins.
In cells from yeast to man, the 2 major drivers of actin assembly, formins and the ARP2 complex, are actually competing for a limiting pool of actin monomer. And so we hypothesized that if we used an inhibitor of the ARP2-3 complex to collapse that network, that these formin-generated structures would get much more robust. And that's exactly what we saw. And then we showed that if you actually added a pan-formin inhibitor in there, you block the formation of these arcs. Beyond that, we were able to show that there are at least 3 different formins that appear at the tips of these linear actin structures, which give rise to the arcs, and that RNAi inhibition of one of them, the one that's most highly expressed, MDA1, leads to a very dramatic reduction in the number of arcs at the synapse.
But how are the formin-generated filaments organized into concentric arcs as they emerge from the DSMAC into the PSMAC region of the synapse?
And the key observation was that when you inhibit myosin II with paranitroblevastatin, the concentric arcs disappear, and that these linear filaments now basically wave around in the medial portion of the synapse with no clear organization. And so what we think then is that the bipolar filaments of myosin II grab these linear formin-generated actin filaments and orient them into antiparallel concentric structures. And this idea is very appealing to us because as these filaments begin to bend, they're going to stochastically bend one way or the other. And so commonly, bipolar myosin II filaments are going to encounter these formin-generated filaments of opposing polarity. And of course, that's exactly what you get in a sarcomere, in a muscle. You need actin of opposing polarity for the bipolar filament to then cause them to slide past each other in a contractile event.
Contraction of the actomyosin arcs, along with the inward flow of the outer branched actin network, promotes immunological synapse maturation by somehow moving T-cell receptor microclusters to the center of the synapse to optimize downstream signaling, and concentrating integrins into a surrounding ring that forms an adhesive gasket with the antigen-presenting cell.
And what we showed is that if you crash the formation of these arcs by inhibiting formin, the centralization of T-cell receptor microclusters and the formation of this integrin ring is abolished. And if you inhibit myosin 2, which isn't required to make the arcs, but to organize them into concentric contracting structures, you phenocopy the effects you see with formin inhibition. So you also get a defect in maturation of the synapse.
Consequently, inhibiting formin or myosin 2 impaired T-cell receptor signaling and adhesion to antigen-presenting cells. But how do the actomyosin arcs move integrins and T-cell receptor microclusters into position?
So in sort of the final portion of the paper, we looked in 3D Sim in a synapse which is in the midst of maturing, and we followed actin, myosin 2, microclusters, and the open active conformation of LFA, which is the major integrin in the T cell. And what we saw from a statistical analysis of many images is that on average, there's an actin arc, which is decorated with myosin 2 and with the open active LFA, and then in between that arc and the next arc is the microcluster.
These static images suggested that T-cell receptor microclusters might be swept towards the center of the synapse as the actomyosin arcs contract and move inwards.
And so to actually see that, we then went again to TurfSim, and that is in fact exactly what you see, that the arcs appear to be essentially sweeping microclusters in towards the center. And so they basically directly propel the motion of these microclusters. Down the road, I think there's lots of other interesting questions to ask. For example, is it really just sweeping, like a broom sweeping dirt, or are there physical interactions going on between the T-cell receptor microcluster and the actomyosin integrin-rich structures? And along those lines, one of the things we're focusing on is myosin 18, which is highly expressed in T cells, which co-assembles with myosin 2 into these bipolar filaments, and which presents protein interaction domains which could possibly interact with microclusters or integrins to help drive this mechanism.
For now, though, you can learn more about how formin-generated actomyosin arcs propel T-cell receptor microcluster movement at the immunological synapse in the paper by Murugesan et al., published in the November 7, 2016 issue of the Journal of Cell Biology.